In this video, we're going to look at post-transcriptional regulation in eukaryotic cells. So this is the regulation that happens after a gene has been transcribed into RNA. And there's several levels at which regulation can occur. So if we take a look here, we are now in the cytoplasm. We've got our messenger RNA here that has been processed and is now within the cytoplasm, and you can see that we've got translation that can be regulated, we've got the degradation of messenger RNA that is regulated, the processing of the polypeptide can be regulated, and even the degradation of the protein can be regulated. So all of these represent different ways in which gene expression can be regulated that is different than the regulation of transcription. So one of the most important forms of post-transcriptional regulations that we are beginning to learn about is regulation via microRNAs. All right, what is a microRNA? A microRNA is a protein complex with a small RNA molecule that is reverse complementary to a specific messenger RNA. Okay, so what we have are these complexes that float around within the cytoplasm, and anytime they find a messenger RNA that they are reverse complementary to, they bind it. And one of two things can happen. Either they actually trigger the degradation of that messenger RNA, or they simply inhibit ribosomes from binding that messenger RNA. Either way, no protein is going to be translated from that messenger RNA. And this form of regulation, where these microRNAs are produced by the cells to inhibit translation of specific messenger RNAs, is very common. In fact, 1/2 of all human genes seems to be regulated this way. Now, the processing, the transcription and processing of a microRNA is fairly complex. Let's take a look at it. So here is a RNA. Okay, this is an RNA that is going to encode a specific microRNA. So this is an example of an RNA that's encoded within a gene that won't go on to encode protein, just like transfer RNAs and ribosomal RNAs and small nuclear RNAs. There's lots and lots of different RNAs that are encoded by genes that, when transcribed, act and function as an RNA. This is one of those cases. Now here, within this big, complex RNA that has been transcribed from a gene right here, this is in orange, is the microRNA that is ultimately going to bind to that protein complex and inhibit translation of some messenger RNA. So first, it's part of this hairpin loop, and drosha within the nucleus cuts that hairpin loop. Then we have another enzyme called dicer, and what dicer does is it cuts off the loop part. So now what we have essentially is double-stranded RNA. One of those strands is called the passenger strand. That is not the microRNA. And one of them is called the guide strand. That is the microRNA. So there is this protein complex called RISC. It recognizes double-stranded RNA and binds to it, degrades the passenger strand, leaving behind the microRNA. And now, within the cytoplasm of the cell, wherever this complex encounters a messenger RNA that is reverse complementary to this guide strand, they'll bind and either degrade that messenger RNA or block translation. Now, researchers, maybe a couple decades ago, discovered this system, discovered that almost all cells have drosha, have dicer, have this RISC complex. And what they did is used these proteins, these enzymes, used the natural ability of processing microRNAs to force the cells to make microRNAs of the researcher's choice. So let's say a researcher wanted to knock down a particular messenger RNA and see, well, what happens when we get rid of this protein that's encoded by this messenger RNA? They inject a hairpin loop RNA into a cell. The cell's endogenous dicer takes advantage of this and cuts that loop off. The endogenous RISC binds the double-stranded RNA and makes a microRNA, and then that knocks down or degrades the messenger RNA that the researchers specifically targeted. This is called, when you use it this way as an experimental technology, it's called RNA interference, or just RNAi. Here's an example of an experiment where researchers did just that. They were interested in a gene called bursicon within silkworms, and they wanted to know, what does this gene do? What does the protein encoded by this gene do? So what they did was they made double-stranded RNA that is reverse complementary to the bursicon mRNA. Now, they had a couple controls. First, they injected embryos, or they injected larvae, with the solvent in which the double-stranded RNA was dissolved. So this is a negative control. They wanted to make sure that simply injecting with these larvae just didn't cause them to generally be sick. And the females looked normal, and the males looked normal. Then they injected larvae with double-stranded RNA that has nothing to do with bursicon. Again, it's negative control. They wanted to make sure that simply injecting any RNA didn't cause these larvae to become sick. And sure enough, females looked normal, males looked normal. Then they did the experimental. They injected larvae with this double-stranded RNA that one of the strands, the guide strand that will eventually become a microRNA, is reverse complementary to the bursicon messenger RNA. And they injected larvae, and sure enough, all the females, all the males showed a very consistent phenotype, a very consistent trait of these short, stumpy wings when they developed into the moths. And so now they can begin to hypothesize, the bursicon protein has something to do with wing development. Maybe they are involved in the growth of the wing cells, or maybe the mitosis, the cell division of wing cells. So this is a common way in which RNAi, or RNA interference, is used within research. Now, there's other forms of post-transcriptional regulation. The regulation of mRNA degradation that's independent of these microRNAs is dependent on 3' UTRs. And in fact, sometimes often the sequence to which microRNAs bind is within 3' UTR. The regulation of translational initiation is dependent upon sequences of the 5' UTR. Proteins, once are made, still have to be chemically modified, like phosphorylated, or sometimes they have to be cleaved in order to be active. And then there's the regulation of protein degradation. Now, another way to regulate gene expression is in the regulation of RNA processing. One of the big and most important forms of RNA processing that's regulated is that of splicing. Remember, splicing is when the introns are cut out of a messenger RNA and the exons are spliced together. Okay, so here then is our primary RNA transcript before splicing occurs, and then as a result of splicing, the introns are cut out, that's the pink areas, and the exons are spliced together. But it turns out that depending on which cell a gene is expressed, can sometimes determine how it's going to be spliced. And some cells splice messenger RNAs differently than other cells. So for example, in cell 1, the troponin T messenger RNA might be spliced in such a way where exon 4 is left out. So now we have a messenger RNA that's only exon 1, 2, 3, and 5. That's going to give rise to a protein that is distinctly different than the alternative here. So cell type B now expresses the troponin T gene to make our primary RNA transcript. But then when it splices it, it leaves out exon 3 but includes exon 4. So we end up having exon 1, 2, 4, and 5. the protein derived or translated from this messenger RNA is going to be distinctly different than this one here from cell type A. So alternative splicing allows the organism to have more different sorts of proteins than genes, because a single gene, depending upon how that messenger RNA is spliced, will give rise to different proteins. Now, proteins will have overlapping similarities, but will be distinctly different. So now we're done with this idea of how the information within all of your DNA, your genotype, is translated into all of your traits, your phenotype. Not only did we look at the mechanics of that, the mechanics of transcription, the mechanics of translation, but we also looked at the regulation of this. And we observed that in multicellular organisms, that it's very important that some cell types turn genes on and others off. And that the way in which a cell develops and specializes is dependent on this idea of differential gene expression. Furthermore, we've looked at experiments that prove that every cell within the human body has all of the DNA necessary to direct the development of a full organism, and that really what makes two cells different are the activators that those cells express, and therefore the genes that they turn on and the genes that they turn off. And all of that goes in to determining who you are, what you are, all your traits. So the next step now is to start to put together the tools necessary for us to understand inheritance. And to get there, we need to first talk about mitosis and DNA replication, and then we'll get to meiosis and Mendelian